Hello, I'm Randall Hoffman, a graduate student in Susan Lindquist's laboratory in the biology department at MIT. Today, I'm going to introduce you to Semi-Denaturing Detergent-Agarose Gel Electrophoresis, or SDD-AGE. Our lab uses this technique to screen for prions and other amyloidogenic proteins. Our version of SDD-AGE is simple and reliable and can be scaled to accommodate any number of samples. Amyloids are exceptionally resistant to denaturation by SDS, which allows them to be separated from other proteins using SDD-AGE. However, data from SDD-AGE cannot by itself be used to confirm that something is amyloid. To do that, you need to use other techniques. With that in mind, let me show you SDD-AGE. SDD-AGE can be performed using standard equipment for horizontal DNA electrophoresis. First, you need to assemble the gel casting tray. The size of the apparatus will depend on how many samples you have. For a large number of samples, we use a 20 by 24 centimeter slab and up to 4 50-well combs. To prepare the gel, you will need medium or high gel strength, low EEO agarose, 1x TAE buffer, and a 10% stock solution of SDS. Make a 1.5% agarose solution in 1x TAE. You will want to make the gel as thick as possible so you can load the most sample and maximize detection. Heat the mixture in the microwave until the agarose is completely dissolved. Quickly add the 100x SDS stock to 0.1% and swirl to mix. If some agarose solidifies during this step, redissolve it on a hot plate. Be careful not to let it boil after adding SDS, as the solution will easily bubble over. Pour the solution into the casting tray while it is still hot. Then use a comb to get rid of any bubbles, which may interfere later on during gel transfer. After the gel is completely cooled and solidified, carefully remove the combs and place the gel into the tank. Completely submerge the gel in 1x TAE with 0.1% SDS. You are now ready to load your samples. The protein samples used in SDD-AGE are prepared just like in an SDS page, with two important exceptions. First, you should avoid heating the samples, which could denature the aggregates into protein monomers. Second, take care to avoid protein degradation, because the denaturing conditions used here are not sufficient to inactivate all proteases. We recommend using at least twice the recommended concentration of a complete protease inhibitor cocktail. In this demonstration, we will use lysates from yeast cells that are overexpressing a particular protein of interest. We generally use 2 ml overnight cultures grown in 96 well blocks available from VWR. But when analyzing low abundance proteins, larger culture volumes must be used. Harvest the cells by centrifuging the culture at 2000 RCF for 5 minutes at room temperature. Following centrifugation, remove the supernatant and wash the cells by resuspending in water and centrifuging again. After the second spin, remove the supernatant and resuspend the cells in spheriplastic solution. The zymal lyase in this solution digests the cell walls, which will allow you to lyse the cells efficiently in a small volume of lysis buffer. Incubate for 30 minutes at 30 degrees Celsius. Pellet the spheriplastic cells, this time at 800 RCF for 5 minutes. Remove the supernatant completely and resuspend each pellet in 100 microliters of lysis buffer. Next, cover the block with tape and vortex on high speed for 2 minutes. This helps all the spheroplasts to lyse. Pellet the cellular debris at 4000 RCF for 2 minutes. Carefully transfer the supernatant to a 96-well PCR plate. Typically, we analyze hundreds of samples at a time in our laboratory, so to assist us with this is a liquid handling system. If desired, determine the protein concentration of the lysates. Next, add 4x sample buffer to the samples to generate lysates containing 1x sample buffer. Incubate for 5 minutes at room temperature. Now load the samples into the gel. If desired, save half of the samples for SDS page analysis. Also load molecular weight markers, including pre-stained markers, and a very high molecular weight marker, such as chicken pectoralis extract, if desired. It is important to keep the gel cool to prevent diffusion of low molecular weight proteins, so you may want to run it in the cold room. Run the gel at low voltage, less than 3 volts per centimeter of the gel length. Run the gel for several hours, until the dye front reaches about 1 centimeter from the end of the gel. Because amyloid is not denatured by 2% SDS at room temperature, it will remain intact, while other complexes, including most non-amyloid aggregates, dissociate in the presence of 2% SDS. Thus, most proteins will be monomeric and run quickly through the gel, while the SDS-resistant high molecular weight amyloids will migrate more slowly. After this, the gel will be ready for transfer. Although multiple transfer methods can be used, we obtain the best results using capillary transfer. Since capillary transfer can accommodate any size of gel, it is practical to analyze hundreds of samples. In capillary transfer, a stack of dry blotting papers acts to draw buffer through the gel, depositing proteins onto a nitrocellulose membrane placed between the papers and the gel. You will need to cut a piece of nitrocellulose, such as high bond C extra, to the same dimensions as the gel. You will also need to cut 20 pieces of GB004 and 8 pieces of GB002 blotting paper to the same size as the gel. GB004 is very thick and absorbent, while GB002 is much thinner and has a finer texture, which ensures more even contact with the membrane. Cut an additional piece of GB004 to be used as a wick, and make it about 20 centimeters wider than the gel. Immerse the nitrocellulose, wick, and 4 pieces of GB002 in 1x TBS. In a plastic container, assemble a stack of papers as follows: 20 pieces of dry GB004, then 4 pieces of dry GB002, then 1 piece of pre-wetted GB002. Lay the nitrocellulose on top of this stack. Rinse the gel on the casting tray briefly in water to remove excess running buffer. Then, carefully begin to slide the gel off the tray onto the stack. While sliding the gel off the tray, douse the membrane with TBS as necessary. The extra buffer helps prevent bubbles from becoming trapped into the gel. A transfer pipette works well for this purpose. After the gel has been completely moved to the stack, check thoroughly for bubbles. If any are present, lift the edge of the gel and reapply buffer until the bubbles can be worked out. Put the remaining 3 pre-wetted GB002 pieces on top of the gel. Ensure thorough contact between all layers by rolling a pipette firmly across the top of the stack. Flank the transfer stack with 2 elevated trays containing TBS. Drape the pre-wetted wick across the stack such that either end of the wick is submerged in TBS. Cover the assembled transfer stack with an additional plastic tray bearing extra weight, such as a 500 ml bottle of water. The wick will draw buffer from the trays, and the dry blotting papers will draw the buffer through the gel. Over the course of several hours, this will deposit the proteins onto the membrane. Allow the transfer to proceed for a minimum of 3 hours or overnight. After the capillary transfer is complete, the membrane can be processed by standard Western blotting. Here you see a typical blot of proteins that were overexpressed in a yeast strain that predisposes certain proteins to form amyloid. You can clearly see that some of our candidates form amyloid or high molecular weight SDS-resistant species very well, like those shown here and here. Others, however, do not form aggregates and are completely monomeric, such as these and these. Additionally, you can see that the size of the amyloid polymers differs depending on the protein, which you can see in these 2 lanes. We've just showed you a simple method for analyzing high molecular weight, insoluble aggregates, such as amyloids of candidate prions. We hope you've enjoyed watching our presentation and find this technique useful in studies of amyloidogenic proteins. Thanks for watching, and good luck with your research.